My name is Shyam Patel, and I'm an assistant professor of medicine in hematology oncology at UMass Chan Medical School and UMass Memorial Medical Center. I'm also a clinical cancer researcher at the UMass Cancer Center. My research focuses on understanding the disease biology of myelodysplastic syndrome and acute myeloid leukemia. Acute myeloid leukemia is an aggressive blood cancer. There are about 21,000 new cases of acute myeloid leukemia per year in the US, and there are about 11,000 deaths per year in the US from acute myeloid leukemia. One of the genes that is often mutated in myelodysplastic syndrome and acute myeloid leukemia is P53. P53 is a tumor suppressor gene, and AML patients with P53 mutation have exceptionally poor outcomes, and there's a significant unmet medical need to develop optimal therapies for this set of patients. For years, it's been well known that P53 aberrations have been associated with adverse outcomes for both myelodysplastic syndrome and acute myeloid leukemia. P53 mutant myeloid neoplasms is arguably the largest black box in the field of hematologic malignancies. Currently, there are 9 Food and Drug Administration approved therapies for acute myeloid leukemia, but there are no Food and Drug Administration approved therapies for the P53 mutation for acute myeloid leukemia. The leading pharmacological compound in late 2020 had been APR-246 or epranepopt from Apria Therapeutics, but this compound failed to meet the primary endpoint in phase 3 data, leaving us with no precision therapies for P53 mutant MDS or AML. For patients with P53 mutant MDS or AML, curative treatment is very challenging, and management plans often include palliative measures or temporizing measures rather than definitive therapy. First and foremost, a deep biological understanding of this mutational subset is necessary to open this black box and to make strides forward. At UMass, here we have transplanted 11 out of 40 patients with P53 mutant MDS or AML, and we found that there was an improvement in overall survival with allogeneic stem cell transplant. We sought to answer the question, what is the basis for the improvement in overall survival with allogeneic stem cell transplant for P53 mutant myelodysplastic syndrome and acute myeloid leukemia? We performed research on this, and we recently published a study where we aim to understand this disease process better. We found that the P53 mutation is a driver mutation for disease recurrence. This is a potentially targetable mutation. We tried to understand the basis for the improvement in survival, so we modeled clonal dynamics by annotating copy number variation analysis against mutant variant allele frequency to infer clonality. We used the inferred clonality to map the mutational hierarchy against clinical outcomes for this set of patients. In summary, we found that both ancestral and descendant P53 mutations contributed to measurable residual disease and fueled disease relapse. Higher mutant P53 gene dosage predicted a low durability of remission. The median overall survival was about 280 days, and survival was improved with allogeneic stem cell transplant. Hypomethylating agent-based therapy served as an effective bridge to allogeneic stem cell transplant, which led to improved overall survival. The high stakes question here is whether or not patients with myelodysplastic syndrome and acute myeloid leukemia harboring the P53 mutation should proceed to allogeneic stem cell transplant as definitive therapy. Formal assessment of the value of transplant has not been performed in larger cohorts of P53 mutant myelodysplastic syndrome or acute myeloid leukemia, and the field does not really have longitudinal data. There is still much uncharted territory, but there is clearly a high need for precision medicine-based approaches for this subset of patients. As longevity improves and as the population ages, it's likely that the incidence of P53 mutant acute myeloid leukemia and myelodysplastic syndrome will rise, because with age, there's an increase in the mutational burden in the aging hematopoietic compartment. The failure of APR-246 was a disappointment to us, but we do look forward to other clinical trials for this subset of patients, including anti-CD47 antibodies, anti-TIM3 antibodies, and Nutlin analogs. These are all in the pharmacological pipeline. Bright horizons might not be too far away, for this subset of patients who deserves much better care. To learn more about our study, please visit our Cancer Center website.